Hello, I'm Stephen Brown. I'm a professor of physics at the University of Warwick. Okay, so my main research field is applying solid-state NMR to pharmaceuticals. I also have an active current research area also applying it to plant cell walls. So within the pharmaceutical world, I've been working with leading companies such as AstraZeneca and GlaxoSmithKline and Pfizer for a number of years now, and the real interest is when you take a tablet, like a paracetamol tablet, you want to know what's the actual specific solid-state forms of that active pharmaceutical ingredient, how they change if you've got humidity, and solid-state NMR is very well suited to looking at them, and also other parts of the tablet, like the excipients, the other ingredients. So in the pharma industry, people know about discovery and know about the drug trials at the end, but there's actually a huge effort in the central part where you found an interesting molecule and you want to get it into a form that can be delivered to the patient. And that's where solid-state NMR comes in that it's a complementary method to other techniques such as diffraction and calculation. So a lot of our work also uses complementary density functional theory calculations. So the other area we've worked in is applying solid-state NMR to plant cell walls. This is a project in particular with Paul Dupree's group in Cambridge, and his father at Warwick, who's still active in his 80s, the founder of the NMR group at Warwick. So I joined in that project with sort of Paul and Ray Dupree, and there the interest so we actually can look at intact, like sort of strands of grass inside the magic angle spinning rotors, and you can see these samples are prepared in carbon-13 labeled carbon dioxide. The plants are grown for a couple of weeks. And you can do 2D carbon-13 spectra that tell you about the actual plant biopolymer and the cellulose, the xylose, the mannose, how they're arranged. So probably one of our key findings was a paper from a few years back in Nature Communications, where we showed that the xylose changes its from a three-fold arrangement to a two-fold arrangement when it goes from solution to being attached to a cellulose, and there you see a big change in the carbon-13 chemical shifts, and those experimental changes were matched by calculations. So the biochemist Paul Dupree in Cambridge had his kind of theories that this is actually what was happening, and then we got the experimental evidence that proved yes, that is what's actually happening. So really, yeah, with NMR, you've got you kind of zooming in on the what's going on around the atomic nuclei up to about 5 angstroms. So really, very close atomic structure, and knowing structure and dynamics in a range of organic molecules. From that one I can say, because one of my roles at Warwick is I'm director of the Warwick Analytical Science Centre. So as that, I get to see the other analytical science methods, and in particular my own research. Obviously for solid-state characterization, the gold standard would be single crystal X-ray diffraction, but there are many cases where you cannot actually get a crystal structure. So in those cases, you're relying on complementary techniques, so powder diffraction, and also calculation. So a lot of our work in the field of NMR crystallography brings together experimental characterization via the solid-state NMR and also the complementary diffraction with calculations where you have a structural model, a solid state, be it from a simulation or a crystal structure prediction approach. So yeah, I always see that solid-state NMR is complementary, and it's interesting to observe. I've worked closely with industry for many years, and there you see that the industry will have experts in all of the relevant analytical science methods, and they'll work together. At the UK, we have a national facility in Highfield solid-state NMR, which had been based around an 850 megahertz wide bore, which has been at field at 2008. And the facility was expanded with the first gigahertz magnet that was brought to field in the UK in 2020. So that's been running as a national facility since 2021. And we're very pleased that, we've secured the funding, and the order was placed in January of this year for the 1.2, with the plan being that it will should come in early 2025, so another year and a half away, but we're still very much actively involved in planning the building, so in the plant cell wall work. So in my talk at this Euromar conference yesterday, I included some results showing the exquisite resolution you get with the gigahertz spectrometer of all the carbon lines for the different biopolymers inside the plant cell walls, and also a recent publication from one of my PhD students working with Pfizer showing how the resolution in the standard proton double quantum experiment for the pharmaceutical lorlatinib, which is an anti-cancer drug, is much better at the higher fields. So yes, we're excited by this availability of these high fields. Yeah, it's always that big question as to where's the difference between, as you go up in field from so Warwick, we've got in the national facility, we have the 850 megahertz wide bore and then the gigahertz and the 1.2 gigahertz coming. A big area of importance is actually quadrupolar nuclei. It's a big area of interest for many of my colleagues in the UK looking at inorganic materials for energy applications and battery applications. And for a quadrupolar nuclei, you kind of get an extra win. So you always win in sensitivity, how it follows the magnetic field to the 3/2 power, and you improve resolution. For quadrupolar nuclei, you're always fighting against this second order quadrupolar broadening of the lines, which isn't removed by the go-to magic angle spinning technique. And that inherent broadening is inversely proportional to the magnetic field. So as you go up in field, your lines get narrower, which obviously improves resolution, but also improves sensitivity because the peak's not so broadened out. So the quadrupolar nuclei in the solid state is really where the big wins are for the high field, and there are many important applications. Even in my own research area on the pharmaceuticals, one of our interesting experiments is to correlate nitrogen with hydrogen using the 14N nucleus, because that's one where your lines get narrower. So it's a big advantage. Thinking about my own research, I'm aware that the plant cell wall spectra, the spectra that the postdoc Rosalie is working with Ray Dupree and Paul Dupree's group from Cambridge, the spectra we're getting are beautiful in terms of the peaks you can resolve and the fine details, and you want to see, like, sugars are difficult to look at, all the proton chemical shifts are quite close together, so being able to spread them out into the 2D spectrum on the high field instruments is great. So we have a low E field probe that kind of reduces the sample heating and is better for samples that are more biologically real, and that's sort of key for our work for the plant cell wall stuff. Okay, so what's the societal benefits of the research we do with solid state NMR? So again, see, pharmaceuticals almost goes without saying. There's sort of the health benefit, and it's often the case that you can identify a molecule that's beneficial once it gets into the bloodstream. How do you get that sample into the bloodstream? And obviously still the most widely used method is the conventional solid state tablet. And in those cases, yes, solid state NMR is essential for following the stability. So again, a lot of our projects with the pharma companies is about stability of a tablet. They need to be able to show to the regulator you store it in the rainforest somewhere in high humidity and high temperature, and it doesn't change its solid state form. And that's where solid state NMR is very well suited. Moving on to the other area where my research is very active in the plant cell walls. There I would say a main factor is trying to use plant biomass efficiently, and one of the barriers in terms of designing better ways to break down the very insoluble biopolymers into forms that are better biofuels is to know the structure, to know how those complex biopolymers interact together. It's, I think, one of the things I discovered, the more you do in science, the more you realize you don't know. And that's certainly the case in what would seem quite basic things we're still discovering, or we're still trying to find answers to, because there isn't really any other. It's such complex multi-systems. The sort of diffraction doesn't really work. There's no other methods. microscopy is limited to what you can see, so the insight you get from solid state NMR is invaluable. One area we've talked about high magnetic field in terms of future developments. An area of continuing development is going to ever faster magic angle spinning frequencies. So in a solid state, we pack the sample into small rotors and we rely on them spinning very fast. So the technology of those, making the rotors ever smaller, allows them to spin faster. And as you spin to faster frequencies, there are multiple advantages, in particular for a topic that's interested me for many years, since my first postdoc, when I was fortunate enough to use at the time, the fastest 2.5mm rotors, is to improve the proton resolution in a solid state. We're still miles away from what you get in solution, but it gets ever better. And that's really where that development in fast magic angle spinning is driving the field forwards.